OMG. Hi. As you can tell, I am a lot brighter now, and I'm not sure if I like it yet or not. You can definitely see my face better though. La 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 la. I don't think I have anything else to say. Um, I hope that my stream doesn't disconnect like it did yesterday. I usually like to do a little bit of talking before I get started on my studying. I've actually done a lot today. I've been awake. No, I actually slept in today. And I missed a brain and behavior lecture this morning, but that's okay. It's actually the first lecture I've missed, like the first Zoom lecture I've missed this entire quarter, which is kind of a miracle. So I'm actually very proud of myself. But yeah, I have an exam for that same class on Friday, and so that's what I'll be studying today. I am practicing active recall questions from basically all of the lectures that we've gone over, and I think that's going to be about it. I already took a stats exam, so I don't really need to worry about that this week. And so, yeah, I don't have a lot of story times for today. I did record a lot of I had a video class today, so it's called Intro to Video and Time Experimentation. And so we actually had to record 5 different scenes, and then the entire class thought that we had to record 2 minutes worth of each scene to get to a 10-minute video. And then it turns out that our professor actually wanted us to get all of that footage that we got and then cut it down to 1 minute. And I was just like, what? We were really expecting to create like a 10-minute video. But thankfully, I was able to figure it out. It was actually lots of fun. And if you guys are interested, maybe I can post a link to the video that I created. It's super wonky, doesn't have a lot of, doesn't have like a purpose or anything, but it was fun to make. So yeah, maybe I'll post a link. I don't know yet. Without further ado though, I'm gonna get the Pomodoro started. I started doing that yesterday, and I kind of liked it, except I had some stream issues, so it kind of messed up the Pomodoro. Hoping that doesn't happen tonight. And so yeah, without further ado, I'll get it started. And then if you missed yesterday's stream, I'm using Momentum, which is a Google Chrome extension, and they have an integration for the Pomodoro in their Plus subscription, and I've been a subscriber for like well over 2-3 years now. Whoa, that's new. Sorry, I have a humidifier running for my plant, and there's a water dot. Anyway, I'm so sorry. So yeah, I'm gonna get started on that. I'm so distracted now. Okay, starting in an hour and a half. And then I apologize for any sniffles. It's quite cold right now. It's a little warmer today than it was yesterday, thankfully, but it's still pretty chilly. Okay, so yesterday I studied neurons and glia, I studied resting and action potential, and then I studied action potentials, and I think I left off on synapse, and I was like dying, so we'll see how that goes. Yep, I didn't even finish it. Okay. You know what I'm wondering too? I'm wondering if the stream ended yesterday because I started using Google Chrome. I don't have a whole lot of apps open so that my computer can just focus on the stream, but maybe. So I wrote that this was my first time going through it yesterday. It was actually my first time going through it today too. And I didn't get very far, so let's see if I remember. Okay, how does intensity relate to action potentials? Oh, sweet Jesus. I'm so sorry. Ah! Okay. How does intensity relate to action potentials? Okay, so, oh, that's right. This had to do with, oh, hi. Okay. Great, now you've distracted me. Thank you. Okay. I remember going through this yesterday. Intensity is basically, oh crap. I remember the slide. Intensity. Oh, the stronger the simulation, for the action potential, the stronger the action potential will be. So that means that the action potential will travel longer and further distances than if the intensity of the action potential were minimal. Okay, so I think that's correct. Let me think about this. It is intensity. It's more intense. The greater the action potential, further it will travel. Yeah, I should be right. Right, coding by frequency. So it is another name for this result. Okay, coding by frequency. This one is coding by frequency. The other one is labeled line coding, which has to do with the fact that there are different types of neurons that receive specific things. Okay. That was correct. Yay! Yellow orange. I make it orange because I totally forgot about frequency. Coding by frequency. This one is coding by, it's called label line coding. Frequency coding? Coding by frequency. Coding by frequency. Okay. What factors determine the quality of action potentials? I remember we're talking about quality. Quality. Give, please. What? Oh, hi. Oh, let me know if you like the new lighting. It's brighter now. I don't know if I like it or not. You can see my face better, but I don't know. Let me know. Quality. Labeled line coding. Labeled line. This has to do with, I believe it was color, shape, and size of something. I think of the stimulus. I think that's what that was. Different types of stimuli qualia activate different separate populations of receptor neurons, such as those receptor neurons used for color, shape. Okay, that's really bothering me. Such as those receptor neurons used for color, shape, size. Receptor neurons. So I did say the right things, but how does that relate? Hold on. I have so much to cover before Friday. My God. it's not this one. It's this one. This one. So stimulus, for example, the sun. No, just the size of the stimulus. The color, shape, and size of the stimulus. So their population is a receptor for that. I mean, I guess. I'm not really sure. That'd be a good question to ask a professor. Too bad I'm not going to. I'm gonna put this as orange as well because I actually got the name right, and then the stuff that it is stimulated by. How does timing affect action potentials? Timing in general is just very important. Oh, whether two action potentials fire at the same time. That I didn't consider, but okay, I'll leave that as orange. Drugs that affect the action potential. Oh, this has to do with fugu. How do local anesthetics disrupt action potentials? So this has to do with general anesthesia, topical anesthesia? No, not topical. I think it's just general. And then this has to do with how the way that anesthesia works is that it actually blocks action potentials from occurring and sending signals to the brain about pain that you are experiencing. So it disrupts action potentials. Of course, that's why you always have to be careful about how much anesthesia a person receives, because it can be detrimental. Right. Okay, so that is related to action potentials. So the anesthetics, they block the voltage-gated sodium channels, which are the gates that open when an action potential is occurring. Okay. Yep, if given too much, you can die. Action potentials stop occurring, hence basic brain death. That's so scary. Target to block. Oh, yay, I'm glad. I'm seeing your messages super late. I still haven't figured out how to get alerts when people put messages into the chat. I don't get any, so I'm sorry. That's why I try to check in every now and then. But thank you. Maybe you can help me with that later. Okay, target. Oh, target is to block. It's like target cis. Target is to block. Plugs the channel. It's very good for blocking impulses such as pain. I got all of those correct. I will make you yellow. Beautiful. How does tetrodotoxin disrupt action potentials? This is the one that's found in the fugu, the fish. So like fugu is like this very special type of sushi that it can be consumed. See, Jesus, I can't speak English. And it's a delicacy because it's poisonous. It's okay, but it's okay because if you accidentally consume it, if you get to the hospital fast enough, they can help you and you won't die. I believe it has something to do with breathing, because from what I remember, they have to put you on a ventilator. Was it a ventilator or was it I think it was a ventilator. It has something to do with the lungs. I think it stops you from breathing or something. Okay, let's see. Aside from that, it's not only found in the fugu fish, but it's also found in red salamanders. So don't eat them. And fugu is another word for the puffer fish. So it also blocks the sodium channels. Okay. Oh, there it is. It paralyzes your diaphragm, which stops you from breathing, and you can be saved by getting put on a ventilator. There you go. So the diaphragm, if you want to know where the diaphragm is, basically you have your lungs, which is like the two, I should like talk to the camera. If you want to know, well, obviously your lungs are like right here. It's like giant lungs, I think. I could be wrong. I'm probably wrong. I think lungs are like right here. And then right underneath that is your diaphragm. If you're a musician, you usually use your diaphragm a lot to play an instrument well. But yeah. Yeah. Blowfish sushi. It's a delicacy. A lot of people like to eat it. I think it costs quite a bit of money too because it needs to be very well prepared. Okay. So I got majority of that correct. I'm gonna leave it at yellow. So I can review it at least 3 more times and then it will ingrain itself into my memory. What do potassium channel blockers do? Oh. Potassium channel blockers do. Okay, so in terms of action potentials, if the potassium channels are blocked, then the membrane can't reach a polarized state again, right? I'm pretty sure, but I don't know what that actually does to the body. It would just be the neuron in a state where it's so excited that it wouldn't be able to restabilize again. Because like the sodium potassium pump is constantly bringing potassium in, and then the channels remain open, so then potassium can also get out if it wants to. What does it do? I don't know. Oh, okay, so I was right. Prolonged action potential. Oh, okay. So fun facts. Scorpions, they trigger action potentials, which is why you experience a lot of pain. And that's usually blocking the potassium channels. So scorpion bite and honeybee stings block the potassium channels from, I believe, opening and closing. So it just blocks them, it like shuts them down. So then it prolongs the action potential, which is dangerous for the neuron, but it also explains why those stings are so painful, because it's so much stimulus from the action potential that it's triggering pain to your brain. Wow. So I was like halfway correct. I just didn't remember the examples. Cool. Demyelinating disease, multiple sclerosis. Wow, we're learning so much today. What are the characteristics of MS? So multiple sclerosis, I went over this yesterday. It is a demyelinating disease. So basically your neurons, some of them on the axons, they have myelin sheaths, which are like bumpers, and they basically help the messages to get across the axon as quickly as possible. So multiple sclerosis, right? What are the characteristics of MS? Basically, the immune system attacks the myelin, which is bad for the axon because then messages can't be sent out as quickly. It was a really long explanation. Let's see if I got it right. Yes, it break down myelins in the CNS and PNS. It's progressive and incurable. Yep, it's very sad. It's one of those invisible diseases. You don't really know when a person has it, or, well, I mean, you can. Maybe once it's advanced enough, you can definitely tell. Causes block of action potentials and thus blocks information transfer. Yeah, because the action potentials can't function as well as they could because the myelin's been taken away. So what are the characteristics? This I did not know. Loss of sensation, which I guess makes sense because action potentials are messages that are being sent. So loss of sensation makes sense. Blindness, I guess if the myelin in your optic nerves is being eaten away at, blindness, all depending on which area of the brain is impacted. True. Okay. I would say I got about half of that correct. I just didn't remember the examples, so I'm gonna give myself an orange. Just come back to that. What are the possible causes for MS? I don't know. I think it's hereditary. It's genetic. It's an immune response. I don't know. Possible causes. Oh yeah, immune system attacks. Yeah, it's an autoimmune disease, which can be genetic. Oh, viral cases. Okay. What are the possible causes for MS? When the immune system attacks myelin, causes inflammation, which is bad for the nervous system. Autoimmune disease, which can be genetic. And there could be viral causes. We don't know why this happens. It's a good answer for everything. So sad. I do not wish MS on anyone. I can't even imagine what they go through. Oof. Oh, but then there is like stem cell research being done to help with this problem, which I learned about before. Okay. I want to explain everything to you guys, but I don't have to. I need to just study. I'm gonna put an orange for that one. What does demyelination do for the axon? So this I was trying to explain earlier, but I was like, I'm getting into too much detail. Okay. So the myelin, okay, think about an axon. So the reason why is because on the axon there are bumpers, which I call them the myelin sheath bumpers, and then in between are nodes of Ranvier. The nodes have sodium channels, whereas the myelin does not have sodium channels. And because the myelin does not have those sodium channels, then basically what happens is the action potential can't move through the axon. That's what happens. There are axons that don't have myelin, and for those, there are sodium channels throughout the axon. But in demyelinated axons, wherever those myelin were, there's no sodium channels there. So yeah, that happens, and it's bad. Did I get it right? Yes. Myelin areas in an axon do not contain sodium channels. Therefore, action potentials bounce off the nodes of Ranvier, but without the myelin, the action potential eventually dies out from one node to the next. Yep. It's very sad. That must really suck for the body in general. Okay. Why do we need myelinated and non-myelinated axons? This I remember as well. We need the myelinated axons for areas where information transfer needs to be as quick as possible. For instance, the brain, but also places like the heart. And then we have non-myelinated axons because there are areas of the body that messages don't need to travel as fast. There might not be a lot of energy resources as well, something like that. And that's, I was talking about, for instance, our fingertips. We tend to, like if we're, I don't have a mug, pretend this is hot, then when it's warm, our hands receive that message quite slowly, because you don't really need to process that information so quickly. Some distances are shorter, or information doesn't have to be quick. I didn't remember the shorter part, but it's why there are. Okay. I will give myself a hello. What are the possible treatments for MS? Is it curable? Possible treatments for MS include, I believe it's just pain management. and if it's curable. It's in the works, but they're trying to use myelin from the peripheral nervous system and the central nervous system to create bridges for the myelin to regenerate itself, because it won't regenerate in the central nervous system. I think that's about it. Okay, so steroids and immunotherapy can help decrease pain and inflammation. Is it curable right now? Unfortunately, no. Steroids and immunotherapy. Steroids, I think that has to do with pain management, steroids, and then immunotherapy. I wonder, what does immunotherapy even consist of? I kind of want to look that up. Let me look that up. What are some examples of immunotherapy? It says by activating or suppressing the immune system. Synapse. Alright, Sherrington's imprints of the synapse by synaptic delay. Okay, so Sherrington was the guy with the dog. Basically, this guy, his dog, he was noticing whenever he would squeeze the paw of the dog, the response that the dog would have, and like how long the delay was before the dog reacted, something like that. What was Sherrington's contribution to discovering synaptic delay? He figured out that there is a synaptic delay. No, that's not the answer. Think about it. Come on. Try to remember. Oh, it was that, wait, was that him? It was something about how, I think that there's like chains of messengers. No, I don't think he discovered that. So as we know it, action potentials can communicate to other action potentials. No. Neurons can communicate to other neurons to communicate to other neurons until the message is finally delivered or not delivered because the stimulation wasn't strong enough. And that's what Sherrington started. Synaptic delay. He discovered that that's just synaptic delay. Synapses. I think he discovered that it takes multiple synapses to deliver a message. So the message has to go through several. I know that he was trying to time it to see like how long or how much time it takes for the message to be communicated. That's as best as I can do. When Sherrington measured the time delay between paw, pinch, and withdrawal, he found it was longer than he had hypothesized, mostly between 50 to 75 milliseconds. Sure. We don't actually need to know this. We just need to know like what he did. It's just measured the time delay, synaptic delay, time delay. Between 50 and 75. think it wasn't miles per second. Meters per second. Because the guy was British, they didn't use the imperial system. Wait. Okay. I'm gonna give myself an orange because I didn't really know that. What was his conclusion? I don't know. Oh, okay. So that's kind of what I said. I just wasn't sure. He concluded that there were synapses in the pathway, C to D, D to E, and that activating a synapse delays the information along the pathway, activating the synapse. Oh my god. Okay. I'm gonna give myself a red because even though I knew that, I didn't know that was his conclusion. Okay, I can do it tomorrow. Okay. So it is what I was talking about. One neuron communicating to another neuron, communicating to another neuron. His discovery is that there is a delay because of that communication. Awesome. Okay. Hopefully I get that right next time. Ow. My head. Temporal and spatial summation. I love these. I spent so much time trying to figure these out. Temporal summation. Okay. So going back to the paw print, I don't know. I like to think of it as a person who's taking a nap in deep sleep. So let's say a friend of yours is taking a nap and you're trying to wake them up. Okay, so temporal summation is, let's say you tap them on the shoulder, right? They don't wake up. So you tap them like multiple times, and multiple times in a short period of time. And then they're like, the fuck, leave me alone, That's temporal summation. And the response is, of course, the wanted response is that they wake up. Now, spatial summation is, let's say it's that same friend. Instead of only tapping them in the shoulder, you're like, all right, I'm going to poke you in the stomach. I'm going to slap your thigh and slap you in the back, give you a slap on the face. Spatial summation. So multiple times in different areas triggers the wanted response, which is for them to wake up or for the action potential to take place. I'm pretty sure that's right, except I'll explain it in a way that makes sense to me and not in the textbook way. Okay, what does it say? The postsynaptic neuron, postsynaptic, pre-post. So postsynaptic neuron is basically the dendrites of the other neuron. Has a threshold, and it needs to be excited, bothered enough to receive the transmission from the presynaptic neuron, which is the synaptic terminals. Axon terminals. That's that area. If there are attempts in succession, temporal summation to excite the postsynaptic neuron, it will produce an action potential. If not, then it fails. Yep. Okay. And then this would be synaptic inputs from separate locations combining their effect on a neuron. A combination of excitations exceeds the thresholds and produces an action potential. Okay, I'm gonna give myself yellow. Because I remembered it correctly, but I need to remember that it's related to to do with the postsynaptic neuron, because although I understand the concept, I need to know where it's actually applicable. Okay. Excitation and inhibition. What is the relevance of excitation and inhibition in relation to synapses? Oh, this is that EPSP, IPSP crap. I don't remember that at all. Oh. Okay, I might be wrong. What? Sorry, this is related to my exam on Friday. What? So apparently someone took a screenshot and it says, make sure you have everything you need ready, because it's open book, basically. Files open, Control F ready. I'm done. And that was from the TA. Control F ready. I'm so dead. Oh my god. And I'm over here actually studying the material. Okay, so I'm studying this material because I actually want to remember it 4 or 5 years from now when I'm in grad school, or even for the GRE, or for like anything. Like, what if I decide I want to become like a neuroscientist in the future? You never know. Like, this is the foundations. Everyone's just like, the fact that she said have Control F ready. I'm flipping dead. Oh my god, I can't get over it. Oh my god. I need to calm down. Okay. Oh my god. Okay. No, this is still on the other side. So like they all want to share a study guide, but that always freaks me out because some professors are hell strict about that. Like last quarter I was taking what was I taking? Industrial organizational psych, and some people basically use the same study guide, and the professor was just like, no, you can't. And like basically like if anyone used that same study guide, that they would get like basically an F on the exam or whatever. So I'm like, I'm so scared to use other people's study guides. Like I'd rather just study it myself. But yeah, okay. No, it's not on this one either. Yeah, it's not this one. I'm done with this one. Get swole. I'm dead.
8 o'clock.
Oh, it's 8. No. So I think it's just that. It's just that. What is the relevance of excitation and inhibition in relation to synapses? That same stimulus could either inhibit or excite specific muscle groups in the body. That is so dumb. That's a very dumb question. I made that question. It's a dumb question. Oh, that is orange. Okay, the Lowy experiment, that has to do with frogs on chemical transmission at a synapse. I have 52 minutes left. Cool. I don't think I like the Pomodoro, because now I'm just looking at the clock. I don't know. Okay. In general words, what is the significance of the Lowy experiment? The Lowy experiment, from what I remember, they were using frog hearts, and they were using a probe. Ow, my head. Oh, that feels better. Okay. I doubt the questions are going to be hard. You guys are distracting me. There was a frog heart, there was a probe, and I think that probe was sending an electrical signal. And I think when it sent the signal, it matched the same wavelength. Something like that. Chemical transmission at a synapse. Synapse. Ow. It's probably making my head look weird. Okay. I think that's my best guess. The probe, the electrical signal, or maybe it was chemicals. I don't think it was chemical transmission. Chemicals? Were they injecting the heart with something? They might have been injecting it and then just testing it. I think that's what they were doing. I don't remember. Let me see. What I wrote is, all I remember is a frog. Experiments done in the 1920s. I did that, I wrote that to make me laugh while I'm studying. That's why I put that there. Using frog heart, you can isolate the frog heart by keeping the vagus nerve intact. So yes, it's in relation to the vagus nerve. When you trigger the vagus nerve, it plays a role in the parasympathetic nervous system. Wow, this is really hurting me. Transferred fluid from the stimulated heart, the heart slowed down just to see the heart. And the vagus shows that chemical transfer messages. This is really hurting my head. I'm not sure why. Transferred fluid. Oh, that's right. Okay. They were using the vagus nerve just to test to see if it was stimulated. So what they did is, so they injected the frog heart on this side with a fluid, and then that showed that the vagus nerve was being stimulated, or whatever, something was being stimulated. And then they took the same liquid they put into that heart into another heart. And yes, that's right. So after they took out whatever they injected in the first heart, the heart began to slow down. And then when they transferred that fluid to the other heart, they saw that then that heart became stimulated, just as the first heart had been stimulated, and then that showed chemicals transfer messages. This was in the 1920s, y'all. Like, this was revolutionary. That's wild. Okay. That is so cool. I did not know this. I remembered a heart and something about a probe. I was right about the probe. Okay, I'll give myself an orange. Okay, the synapse and its structure. Image of a synapse, describe it. Okay, I kind of cheated because I looked at the PowerPoint, so I kind of remember what it looks like. Okay, so he put it really simply for us. There's the presynaptic neuron, the postsynaptic neuron, the synaptic terminal or the synaptic cleft, which is where the so in here are like vesicles, like inside, like this on this side, inside. Wait, well, I can imagine it. You guys can see it. It's like a funnel. So this is the axon. Then you have the axon terminals, and then what's it called here at this part of the membrane, there are like receptors and channels. And then inside here, there are vesicles or small spherical things that carry the neurotransmitters. And so when they pass through these, what do I call them? What are they called? I don't remember the name. I'm just gonna call them receptors. They go through these receptors and they're almost like changing rooms. So, or does that happen in the vesicles? I don't remember. But so like when they're in the axon, they have charges. They're chemicals like potassium, sodium. They have charges, positive or negative. And then when they get into the vesicles, it's like they go through a changing room where they become neurotransmitters, which are different. They don't have a charge just yet. And then they'll go through the same change again in the postsynaptic neuron to get the charge again in order to like communicate with, it's like they change languages. Yeah. And then they're without charge so that they're easier to transfer. Right, so there's that. And then on the sides you'll have glial cells, and the glial cells help to pick up any waste. And then there are other receptors on the edges that pick up empty vesicles and return them back to the presynaptic neuron. And then here's same thing with the presynaptic. Watch me get this wrong. Okay, I'm pretty sure I'm right. So they're like all free in this little space. Messages, neurotransmitters trying to get to the postsynaptic neuron. And then the message is delivered and then they go through the thingy and then, yeah. I think I'm right. I was right. It's much simpler than what I imagined in my brain, but okay. Neurotransmitter transports. Oh. Okay. I might have been wrong. Okay, presynaptic, you have the vesicles, little green things, and then the neurotransmitter transporters. So those vesicles get on the transporters, or they deliver their message to the transporters, and then there's receptors on the postsynaptic neuron. And then glial cells also have neurotransmitters. Fascinating. Okay, I got that majority right. What is the significance of gap junctions and where are they mainly found? Electrical synapses. Gap junctions. That's the changing rooms. The electrical synapses. Honestly, don't remember more than that, and that's just my assumption. So I'm gonna go for it. Okay. Ion channels directly through each membrane. Connexons. So gap junctions are ion channels. I was right. Don't have delays. I remember what they are. Why are they called gap junctions? They should be called gapless junctions. So gap junctions are in a membrane. What is it? Usually I don't know how to describe it. So like a membrane has okay, membranes have receptors, right? And so they let things in or out. Gap junctions, it just goes straight. So it's on either side of the membrane. So the membrane's bilayer has two layers. So there's an entrance and an exit. This one is an entrance and exit all in one. So it just flows right through. They should be called gapless junctions. Channels about Oh, channels abut to each other, giving access to other cells. Yeah, useful for very rapid synchronous transmission, such as in the cardiac muscle. I did not get that right whatsoever. I was still in synapses. Okay. So this is looking okay. Yeah. Yay. Okay. So I plugged in synaptic integration. Did I? Oh, there's EPSV, IPSV. And then after that, I have to plug in neurochem, neuroanatomy, and development, which I have not done yet. So that's probably what I'm going to do after I finish studying the synaptic integration. Once I do that, inputting everything, like plugging in, takes forever. I'm not looking forward to that. Like, I make it look fun, but it actually takes a long time to input everything. Okay. Alright, I'll change this to bad. This is my first time studying this portion. Basic categories of synapses based on location. I remember these. Alright, so we have the first question. Where does the axosomatic synapse connect to? So this synapse connects to the soma, or the cell body of the axon. That's what it connects to. Connects to the soma? Yes, 100%. Where does the axodendritic synapse connect to? This connects to the dendrites. Yay! Good job. Okay. Where does the axonic synapse connect to? Oh, I think this one is the I think it connects to the synapse. Wait. I was so confident. Maybe it is to the axon. Let's see. The axon terminal, which is the synaptic terminal. I was right. Oof. Okay. Oh my god, yellow. You do know what it is, Nancy. You do know. Where does the dendrodendritic synapse connect to? Dendrodendritic. This is a dendrite connecting to another dendrite. That's what that is, I think. On dendrites, not just input elements, they can send info as well, using synapses using dendrodendritic. Dendrites connecting to other dendrites also sends synapses, which is accurate because... Okay, so I'm confused on one thing, and that is that the axon very much looks like a neuron. Wait, no. Okay, the neuron, cell body, this is the axon. This is the axon, that's it? Okay. It's a neuron. Got it, okay. I am not confused. Alright. Neurotransmitter release, exocytosis. What is the process of exocytosis? Well, obviously it's release. I don't actually think I know this. Obviously the neurotransmitter is inside the vesicle, and then it exits the presynaptic terminal via neurotransmitter transporters, and then inside those neurotransmitter transports, my god, that's a long name, they are received by neurotransmitter receptors on the postsynaptic neuron. The transporter fuses with the receptor. I think it fuses. I could just release it. No, I think it like parks. It it's kind of like a port. It docks itself, releases, and then it's inside the other neuron. I think that's it. Oh wow, there's like a 1 through 6. Okay. 1. Oh, that's fine. Okay. This isn't what I said. Okay. Exocytosis. Exocytosis. 1, 2, 3, 4, 5, 6. Okay. Exocytosis. Alright. So, obviously, postsynaptic, presynaptic axon. The action potential has delivered the message to the terminal. AP depolarizes the terminal. Depolarizes terminal? Yes, okay, so the action potential reaches the terminal and it depolarizes the terminal. I didn't even know that had to happen. Okay. That depolarization opens voltage-gated calcium channels. Calciums are double positive. Wow, voltage-gated calcium channels. Okay. Calcium channels flow into the terminal down its concentration and electrical gradients. Okay. Calcium binds to a variety of proteins that ultimately lead to the physical fusion of vesicles to the plasma membrane. Yeah, okay, let me read that one again. Calcium binds to a variety of proteins, ANEGs, that ultimately lead to the physical fusion of vesicles. Right, so we have calcium ions, proteins. So both of these things. I'm assuming that the calcium ion needs the proteins as like food. I could be wrong. I'm probably wrong. And then for some reason, the merging of these two allows this to fuse with vesicles. So vesicles are like little ships inside the terminal. Calcium, proteins, vesicles, they fuse. Yes? No. Calcium proteins help. Okay, the way I'm imagining it is like, what movie is that? I don't think it's Star Wars. No, it kind of is. So like when the ships land in, like for instance, the Death Star, like they have to, like they like dock onto like a thingy that takes them to their parking spot inside, and like they can't like release themselves, like they have to be actually released to dock them. So that's like those docking mechanisms is the calcium and the proteins. And so what they do is they help the floating chips, the vesicles, to bind to the membrane. Okay. I think that's it. I'm gonna have to read that in the textbook to make sure. Right, okay. They have reached the Death Star. Neurotransmitters. Diffuses out of the vesicle and into the synaptic cleft. So the vesicles are literally just to get it to the membrane, and then it releases. Because the vesicles, inside the vesicles, neurotransmitters are already in there. Okay, I need to learn more about the vesicles. So when the vesicle fuses with this membrane, it releases. So inside the vesicle is where the change is happening. Okay. Synaptic cleft, empty space between the pre and the post. Release neurotransmitter binds to receptors to affect targets. Yes, so there are receptors on the postsynaptic neuron, postsynaptic terminal, and then the receptors there, they will bind together to affect, to send its message. Fascinating. Wow. Okay, let's see if I can remember that. So 1, 2, 3, 4, 5, 6. 1, the action potential lands in the terminal and depolarizes the terminal. Good. 2, calcium voltage gated channels open. That allows calcium to release. Step 2, the calcium binds with proteins, and then they dock. No, they dock. They dock the vesicles to the membrane. Yes. Then from there, after fusion with the membrane, the vesicles release neurotransmitters. 1, 2, 3, 4, 5, 6. Right. Then, okay, so they're released into the synaptic cleft, and then, yes, okay, and then the receptors in the post-synaptic terminal, they are received. Wow, took me forever to find the word. The receptors receive the neurotransmitters, and then once inside, they deliver they bind to it. Oh, wow, what's wrong? Oh, so that was the last step. I missed a step, which is calcium flows into the terminal down this concentration in electrical gradients. Other than that, I was right. All right, cool. I am going to put that as orange, because it kind of knew, but not really. What is contained within vesicles? Neurotransmitters. Contains the neurotransmitter, which then bind to receptors to be released. 100%. Good job. I really want to read about this. I'm going to remind myself to read about it. How's everyone doing? Thank you! Yes, I'm trying so hard to study. I appreciate your message. Oh, that's interesting. I can't see it here, but I can see it. I like how you guys are just watching me struggle with new information. With my stuttering and everything. Okay. I hope your studies are going well too. It's not just about me. Oop. Oh. 30 minutes. All right, cool. So I've been studying for like an hour. That's great. Types of neurotransmitters. Okay, what are the different categories of neurotransmitters and their properties? There are categories? Oh my god. Okay. I don't know. Because this is the first time I'm hearing that there are categories. Not really, but I don't remember. Oh, these. working very hard to memorize these. Yep. We have the monoamines, which are single amines, just one amine, monoamines. Mono means one. Then we have amino groups, amino acids. Amino acids, which contain an NH3 group, which is nitrogen and 3 hydrogens, or something like that. And then we have peptides, neuropeptides, which is a chain of amino acids. Single, and then the amino, and then... Oh, I'm wrong. Okay. Monoamines, a neurotransmitter, contain one amine group and NH2, not NH3, NH2. Right, formed by metabolic change in certain amino acids. Right. Then you have amino acids. It's an acid containing an amine group, NH2. And then the neuropeptide is a chain of amino acids. Okay. My brain is getting tired. Maybe I'm due for a break. No. You can do this. We can do this. Acetylcholine. A one-member family. A chemical similar to amino acids, except that the NH2 group has been replaced by an NCH3 group. Yeah, I know, that's a lot. Purines, category of chemicals including adenosine and several of its derivatives. And then we have gases. Nitric oxide, not to be confused with nitrous oxide, and possibly others. Okay, there's 6 of them. Whew. My brain's just like, you want to put more inside your brain? And I'm just like, yeah. Oh, Dad. Okay, someone posted question 1. What is your purpose? A, yes. B, no. C, A, and B, no. Oh, C, A, and B. D, none of the above. I'm dead. That's such an existential question. Oh my God. Ow. Alright, I can focus. It's cold. Can you tell my train of thought is like, okay. I worked so hard to memorize the last thing that I don't want to memorize this new thing. Oh my God. Name some of the types for each of the category of neurotransmitters. Oh my God. I'm really glad that this is going to be open book, because I don't know if my brain could have memorized all of that. It's seriously hurting my head. Okay. Let me close this. Let's see if I can remember them. Monoamines, amino acids, neuropeptides, purines, gases, acetylcholine. think this is going to be one of the ones that I write down. Yep. I kind of already memorized it, but still. Miss Brown, Miss Black. Okay. Oh my God, it's that cold. Okay. All right. Let's see. Categories. Neuro. Transmitter. Categories. My hand is moving so slowly because it's so cold. All right, we have amines. We have peptides, acetylcholine, gases, purines, and CH33 group. I think that's what the acetylcholine was. No, not that one. And CH33 group. Yes. And CH3 group. NH2. Whoops. Tipped. I wrote it so nicely too. Okay, try it again. I'm giving my brain like a little break right now by taking my time to write this. Neurotransmitter categories. Amines. Amino acids. This is actually going to help me memorize them too. Peptides. Crap, that came out ugly. Acetylcholine. Purines. Finally, gases. I don't think I actually had to memorize these. I think I just did this to myself for no reason. Okay, this is an NH2 group, right? And then this is an NCH3 group, right? Another one to add to my little. No, it's not even. Not even either. I'm giving myself a little brain break. Okay. Now that I see it written down, monoamines, amino acids, neuropeptides, acetylcholine, purine, gases. I will never forget it again. I probably will. Watch. Imagine, like, if someone at work were to ask me, like, what are the different categories of neurotransmitters? I'd be like, I don't know. Okay, can I describe them? Monoamines is a single amine group or NH2. Amino acids are an amine group. No, are acids with an amine group. There we go. Neuropeptides are chains of amino acids. Acetylcholine is a what was the word that they use? It's like a manipulated amino acid, and that it has NCH33 groups instead. A one-member family, something like that. Okay, and then purines. Yeah, that one I don't know if I can remember. A category of chemicals, so I guess just chemicals. And then gases is gases. Okay. I knew what they were, but I couldn't remember them, so I'm gonna give myself a red. What are some of the types for each category of neurotransmitters? It's coming back to me. Okay, this has to do with like dopamine, serotonin, what was the other one? Norepinephrine and epinephrine, and then there were a bunch of them. Obviously nitric oxide for gases, purines. I don't remember what they were. Chemicals, could it be like vitamin D, vitamin E, acetylcholine? I don't remember. Okay, I think monoamines was dopamine. I think neuropeptides, I don't remember. Okay. Let's see what I wrote. Yeah, okay. Oh wow, yeah, no, I named all of the monoamines. I didn't name any of the amino acids or peptides. That's a lot to remember. I probably won't remember any of these, and I feel like I should. I feel like I should write these down. My whole desk is going to be lined up with types of neurotransmitters. Actually, I might just do that. Give myself a brain break. I'm sorry. My nose it's really cold right now. This is a mess. Please ignore it. What do I have in here? Oh, I have stickers. My stickers! Oh my god! I've been meaning to put these on my car. I didn't put them on my car. Do you guys want to see? I'll show my stickers later. Stop. I'll come back to you. It's like straight up distraction, my god. Oh. Should I make them pink? Yes, pink. Because my class is pink. The color theme for my brain and behavior class is pink because the brain is pink. Okay. I'm gonna get very decorative. I'll use this one. Okay. Do I have enough? 1, 2, 3, 4, 5, 6. And I have 3 just in case I make mistakes. Perfect. Monoamines. Good. Should I write them in pencil first? Stop being such a perfectionist. Monoamines. Okay. I need to relax. Relax. It's okay. isn't it like this one? It's because it's GS. Slightly better. Every time I say that, I feel like I'm about to say Indonesia, but no. Oh my god, that's ugly. Okay, no. No, I don't like this marker. That's so thick. Okay. Mistakes happen. Switching to my zebra liner. I like that better. Okay. Oh crap. Epinephrine. I need to sneeze. Okay, good. You guys didn't hear me sneeze. Don't need to write that. That's just for me. Okay. Woo! I have my first one. Nice. Cool. Then we have analysis. acetylcholine's. Okay, and then I'm like super quiet. Oh wait, I still have 3 minutes. Okay. It's almost time for break. In like 3 minutes, I'll be taking a 15. Okay. Push through, push through. Okay. Gases. And purines. I think I'm gonna write acetylcholine, gases, and purines in one. Okay. I spelled it wrong, didn't I? Oh, I didn't. Okay, good. Acetylcholine. Then we have purines. Such as ATP. Leucine. Gases. Nitric oxide. Perfect. These sticky notes are so old. They don't want to stick anymore. Okay. All right. Okay. Make this red. We're getting into the information that I actually want to know. Neurotransmitter reuptake. What processes clear neurotransmitters from the synapse? So that's that that change that I was talking about, where like it loses its charge and then it's able to travel into the next neuron, onto the next neuron. I don't know. So that's what we're studying because we don't know. Okay. What processes clear neurotransmitters from the synapse? Uptake by transporter molecules, mostly into glial cells. Cleanup. I think cleanup is a better word for clear in this sense. So any neurotransmitters that I guess weren't used or whatever, because oh, that's right. Neurotransmitters, they make more than they actually need. Yeah. So sometimes when there's just like extra that they don't need, they'll be cleaned up by the glial cells. So there's an uptake by the transporter molecules, and then it's a process of endocytosis or reverse exocytosis and vesicle recycling. Yep. Okay. Cleanup. Glial cells. Recycling. I did not know that. So that's red. Ionotropic and metabotropic receptors. I think these are the changing ones. I keep mentioning them because I don't know when they're going to come up. Okay, ionotropic receptors are receptors that pick up ions or molecules with charges or chemicals with charges. And then the metabotropic receptor. Actually, let me see if I even got that right. So receptors. Okay, so these are the stuff in the membrane. These are stuff on the membrane. There is an ion channel built into it. When a neurotransmitter binds, it opens the channel and ions will flow depending on their concentration and electrical gradients. Where are they found? Directly causes changes in membrane potential. I don't know where these are. Has an ion channel built into it. It's a receptor, so if it's a receptor, then it has to be on the postsynaptic neuron, right? When the neurotransmitter binds, it opens the channel, and ions will flow depending on the concentration and electrical gradients. Oh, directly causes changes in the membrane potential. So I think this is occurring at the postsynaptic neuron. So the postsynaptic neuron has receptors. And those receptors, I'm assuming, are these two different types. So basically at the ionotropic receptor, it receives ions. So I thought they had to go through a change. So when the neurotransmitter, so after it's released, after the vesicle fuses with the membrane and releases the neurotransmitters into the synaptic cleft, the neurotransmitter will bind to its appropriate receptor, and it opens the channels and the ions will flow out of the neurotransmitter depending on their concentration and electrical gradient. So this goes back to cleaning up neurotransmitters. If the concentration or the electrical gradient pushes away the ions within the neurotransmitter, then they won't be used, and then they'll be taken up or cleaned up by the glial cells and recycling receptors. Now, if it is taken in, say by the concentration or the electrical gradient or combination of both, then obviously it will cause a change in the membrane potential of that new neuron, because that's the message it's trying to send. Beautiful. I did not know that. That is red. Okay. What are the properties of the metabotropic receptor? Metabotropic. So I'm assuming a similar process, but this time, what is it taking up? Associated with a G protein inside the cell. When neurotransmitter binds, it activates the G protein, which diffuses away from the receptor and activates any of a large number of intracellular signaling processes. I'm gonna need to study that more. I think I was supposed to write down that I'm supposed to read those two things. Stretch. Read up on. also on vesicles and neurotransmitters. Oh, and calcium. Okay. No! Okay. Wait. I studied 5 minutes into my break. Oh my god. Okay. Oh crap, I went further than I meant to. Alright, so I've been studying for an hour and 40 minutes because, no, 35 minutes because I studied straight into my break because I didn't hear the little ding. Y'all probably heard it, I didn't hear it. So I'm gonna take a break for 15 minutes and then I shall be back. If you've been studying this entire time, you should probably also take a break too. If not, I mean, it's up to you, honestly. Alright.
Nine o'clock.
always forget to unmute myself. Okay, so let's see, if I go for another hour and a half, it'll be just about 11. So I will probably study for 1 more hour and then call it a night. So I should be done by like 10:15. Ooh, display happened. The last few seconds. Alright, so I won't be using the Pomodoro. I think that's what might be triggering, like opening Google Chrome and all that is what's triggering the skipping frames and everything else. But maybe we could have like a few minutes to just talk. So these are my stickers. I have no idea if you find this at all interesting. But I went on a road trip over the summer and it was like the best thing ever. I had the greatest time of my life, and you know, I went to a lot of really amazing places. Like, it was my first time ever going to Yellowstone, and Yellowstone, let me tell you, if you've never been, you need to go. It's beautiful. You see a lot of bison, a lot of elk. Is it bison or buffalo? I think it's bisons. I was kind of sad that I didn't see any animals chasing each other to eat each other for prey, but I used my imagination and that worked just as well. But yes, these are some of the stickers that I got. I went up the West Coast and down the West Coast. I started in SoCal, obviously. Went up all the way to Washington. And originally the plan was to go to Canada, but then, well, COVID closed all borders, so we couldn't go to Canada, but that's okay. We ended up coming back down. We stopped by in Idaho, Montana, Utah, Arizona, and finally made it home. So yeah, this is a Utah sticker. I actually wanted this on my car. And then I went to Zion for the first time and it was amazing. I really, really love this sticker design. Because when we were driving up the coast, like I kept seeing cars with these stickers on them and I'm just like, I love that. And so I tried my best to find stickers that had that design, but they were actually kind of hard to find. And then this is another Utah sticker. Utah has like a lot of really great spots. For instance, the lava hot springs. I went to the lava hot springs and it was wonderful. I had a great time there too, met some really fun people. I went, not water rafting, that's something else, though I have done that in Costa Rica. Water tubing. Yeah, it's basically like Bigfoot Rapids from Knott's Berry Farm, but in real life. And it was lots of fun. It was cold as heck, so I was kind of like a chicken about it, but I really, really had a great time. And yeah, they had hot springs there, so after being in the cold water, I was really happy to get to the hot water. Although, maybe not the best idea to go to hot springs in the summer, but I still had a great time. And then, yeah, oh wait, was that in Idaho? Is this Idaho? This is Idaho, right? I think Lava Hot Springs was in Idaho. I thought it was Utah. My bad, but yeah. And then I did get a chance to go to the Grand Canyon. A lot of stuff was shut down there though, and so that was kind of disappointing. But like they usually have a shuttle service, and the shuttle service was closed, so that kind of sucked. But this is the sticker I got from there. This is something else. And then I don't know if I'll get like copyrighted for this, or I don't know, flagged for this. My video is, you know, not for students, for children. But then I have this sticker too, which is kind of a play on words. It's Salt Lake, Utah. Yeah, I got that. I don't know if I want that on my car yet, but maybe. And then this is my Yellowstone sticker. I kind of wish I had gotten the bigger one, but it's okay. And then I have a smaller version of the Salt Lake, Utah sticker. And then in Arizona, I found one for Best Life Ever from Flagstaff, which is a lovely time. By the time I started going for these stickers, I couldn't get one from Yosemite, from Oregon, or Washington, so that's okay. It's not like I'm not gonna go back up there again. I will. I love to travel, so yeah. Those are my stickers. And then these are just REI. They have a lot of really great outdoor stuff. I'm not sponsored, but I am a member. And yeah, so those are my stickers. I was originally gonna put them on my hydro. Like I have the Hike It Utah one on here because I do really, really love to hike. But yeah, so that's a little peek into my life. Oh, damn it, I dropped the sticker. Yeah. I need to put these on my car. No, I can't leave them there. I need to wash my car though. Yeah, I was gonna put those stickers on my hydro, but I decided not to. Because I really want to put them on my car. Okay, so that was a little bit of story time and kind of a break. Let's just try to get back into it. Probably till 10, and then maybe I'll talk a little bit more, and then I will end the stream. But yeah, let's get into it. Nope, nothing. Okay. Do 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 do. Ba ba da bum. Let's study. Oh, that's right. I have to do that next. No! I forgot I had to input and just study stuff. I really haven't been paying attention in my most recent lectures. I really need to. Because I've just been like, oh, just read the textbook. Which is so stupid. I mean, I will, but usually only look at the textbook for stuff that I'm just like, I need to know more. But yeah, anyway, okay. We left off on ionotropic and metabotropic receptors. G proteins. Okay, let me write that down. G proteins. I don't know anything about that. Really, really don't know anything. So I'm gonna put that as red. All right. EPSP, IPSP, excitatory something something something, and inhibitory something 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 is I think the names for those. How do EPSP and IPSP affect synaptic activation? I don't know. I really, really, really don't know. Okay, so it says excitatory postsynaptic potentials. Okay. Excitatory depolarizing events. Makes sense. Generated by synaptic activation. Makes sense. So I'm assuming that this is a process that occurs once the neurotransmitters have been received by the postsynaptic neuron. And it says postsynaptic. And then if those by the concentration electrical gradient are accepted by the postsynaptic neuron, postsynaptic terminal, postsynaptic whatever, then it either depolarizes or inhibits. Okay, so in IPSP, inhibitory postsynaptic potential are hyperpolarizing events generated by synaptic activation. Interesting. Both cause a transient change in the membrane potential. The exact size and duration depend on the neurotransmitter and channel. Okay, so I definitely need to learn more about those two. And I will study them when I read up on them. So I'm going to make that red. So that concludes. I literally only had that last question and I couldn't finish it before I ended. My knowledge of this is very bad. Yeah, it's bad. So now input active recall questions for neurochemistry, neuroanatomy, and development. Because they be empty. Okay. This is going to involve me opening PowerPoints. But I don't know which ones. Feel like these all have to do with just like directions. Development is the one where it's a doozy. That one I remember like went into it. I'm like, oh my god. That one I'll definitely have to watch like lectures and stuff. Oh wait, before I keep going though, do I have anything due tomorrow? Tomorrow, nothing. Oh, okay. But tomorrow I have classes, and then I have work. Yep. It's crazy. So tomorrow I have stats, and then brain behavior discussion. During discussion, we're probably going to go over questions for the exam. And then the exam is Friday morning. Oh my god. Oh my god. Oof. Oof. What should I do? Can't do an hour or something. I'm gonna have to wake up early tomorrow and study. Oof. OMG. I mean, thank God it's open book. I have a really good grasp, pretty good grasp of the first 4 sections. Oh my god. If I could scream, I would. See, this is why the title of this video is I'm a clown. Because I'm a flippin' clown. Synaptic integration. We're gonna go into neurochem right now. Okay, let's get into it. Let's get into it. I think it's at the end. No, I learned that. Okay. I learned that. That's confusion. It's just an image showing what it looks like. Neurotransmitter reuptake. Yeah, we went over that. That's this. So what I'm gonna do is I'm actually gonna copy this image to my notes, because this is what I was imagining, so I wasn't wrong, but I also wasn't like 100%. There we go. If it's skipping all the frames, I'm so sorry. Synaptic integration. Importance of synaptic. I didn't input these yet. And I think this is because this is the neurochem, but it isn't. This is still synapses. Oh my gosh. Okay, this is for 5 goes straight into neuroanatomy. Yeah. Neuroanatomy is pretty short, and honestly, like, I already know it. I think I do need to study it, but I kind of already know it. So neuroanatomy isn't like a big deal. Development is a big deal. Because that one is a lot. That one, I don't even think I'll be able to get to it. And we're gonna go through all of this. Oof. Okay, so he didn't go over chemistry. Points of synaptic location. Each synapse generates an EPSP of the same amplitude, which yields the largest EPSP measured at the Psyc initiation zone. Recall this is the part of the neuron where it's easiest to trigger an AP. Thus, this is the point at which the output of the cell is controlled. Is this neurochem? In general activity of many synapses requires to generate a spike. SIZ. Synapses inhibition will decrease the probability of spiking or reduce spatial approximation. Oh! I didn't see these. Glial transmission of tripartite synapse. I think this is the neurochem part. Astrocytes communicate between synapses and vasculature. Spinal circuit. Yep, this is it. This is the neurochem part. Where were these slides? Okay. This is still synaptic integration. Astrocytes. This is neurochem. These slides are neurochem. Other neurotransmitters, drugs, synapse, drugs, okay. Yeah. Obviously, this is a brain behavior class, so you're obviously going to discuss drugs and how they impact the brain. Just a good thing. He left off here. Okay. Did I input these? I have a feeling. I'm not sure what I did. Let's see. Okay. That's all I did. What happened? What did I do? What day was this? January 20. What's going on? Wednesday. Last Wednesday. Okay, like I missed lecture today for sure. I did not attend. Okay, this calendar is useless. Oh. Okay, I remember. I was flipping out because of my video class. I remember. Okay. Bad, Nancy, bad. Wow. Okay, so I definitely missed. I didn't pay attention. I attended, I just didn't pay attention. Okay, what shall I do? Make questions. My keyboard is kind of dirty. Okay. How is everyone? Good, good. So this is header 2. I have to like basically type everything out because I didn't do it from my notes, which is bad because now I can't copy paste stuff and just create questions. So with that being said, this might take longer. Semantic application. But it's still okay because as I'm going through it, I'm studying it. I'm just not actively recalling it. So it's not necessarily the same. Alright, what question am I asking? What is the importance of the SIC? Spike initiation zone. Right, and then this is what I want to do. Why is this a question? In general, to synapses, it is required to generate a spike. Yes, the APSP generates synapses, generates amplitude, the same amplitude. Each synapse generates an APSP. This is what I get for missing lecture. Oh no. I have to consult the textbook. Which isn't the best. Well, I mean, the textbook is great, but that means that I have to read stuff. Synapses. Chemical events. There we go. Oh, I remember this. I read about this. Oh, because I was going over the transmitters. Activation of receptors. Ooh, this is the stuff I wanted to read. Ionotropic, azeotropic. So, thankfully, G protein. Oh, no, I'm not trying to learn that right now. Oops. I don't think you guys can see that, can you? I hope not. Well, it has both female and male. Okay, so we are over here. Oh, but this is still about EP. Okay, no, yeah, I need to go back over here. I do actually need to read this then. Okay. Listen to this. The synapse is a complicated place, obviously, with many proteins that tether or attach the presynaptic neuron to the postsynaptic neuron. Abnormalities of these scaffolding proteins, the proteins that hold stuff together, or have been linked to increased anxiety, sleep disorders, and other behavioral problems. Oh my god, what? That's interesting. Their functions are to hold the neurons together and to guide neurotransmitter molecules to their receptors. I'm assuming because those issues involved issues with neurotransmitters not reaching the places they're supposed to reach. Fascinating. In English, return is a kind of mind. In this case, in German, return means far away. In French, the term is meaningless. Okay. Similarly, the meaning of a neurotransmitter depends on its receptors. Wow. Okay. Each of the well-studied neurotransmitters is known to interact with several different kinds of receptors with different functions. Therefore, a drug or a genetic mutation that affects one receptor type may affect behavior in a specific way. That makes sense. For example, one type of serotonin receptor mediates nausea, and the drug ondansetron that blocks this receptor helps cancer patients undergo treatment without nausea. Fascinating. A neurotransmitter receptor is a protein. Yes, okay, neurotransmitter receptor. Okay. It's a protein embedded in the membrane of the postsynaptic neuron. When the neurotransmitter attaches to the active site of the receptor, this is my Death Star analogy, the receptor can directly open a channel. That means take down its, what is that, the wall, the force field. Then when the neurotransmitter attaches to the active site of the receptor can directly open the channel, exerting an ionotropic effect, which we have learned is basically opening the voltage-gated channel that allows the ion to be released, and then it'll trigger or not trigger. or it can produce slower but longer effects, exerting an ionotropic effect. Ionotropic effect, I'm gonna make it equivalent to an action potential, but just like the beginning of an action potential in another neuron. Or it can produce slower but longer effects, a metabotropic effect. Metabotropic having to do with the G protein, which I know nothing about. Ionotropic effect, certain neurotransmitters exert ionotropic effects on the postsynaptic neuron. When the neurotransmitter binds to a receptor on the membrane, it opens the channels for some type of ion. The sodium and potassium channels along an axon are voltage gated, yes. The channels at a synapse are transmitter gated or ligand gated channels. So it's different types of gates, different keys. Okay, a ligand is a chemical that binds to another chemical. Ligand gated, transmitter gated. The transmitter does have to bind to the receptor, which is why it's ligand gated. Okay. Ionotropic effects begin quickly, sometimes within less than a millisecond, and they last only about 20 milliseconds. Because it's an action potential, it comes charged. Ions have charges, it comes charged, so it happens quickly. Okay. Most of the brain's excitatory ionotropic synapse. Oh my god. My brain. Most of the brain's brain's excitatory ionotropic synapses use the neurotransmitter glutamate. Okay. Excitatory glutamate. It ends with an E. Excitatory. Okay. Most of the inhibitory ionotropic synapses use the neurotransmitter GABA. Inhibitory. It has a B. Inhibitory. OMG. Gamma aminobutyric acid. Gamma aminobutyric acid. GABA aminobutyric acid. Okay. Inhibitory. Excitatory. Inhibitory. Okay. Inhibitory ionotropic synapses use the neurotransmitter GABA, which opens chloride gates. Enabling chloride ions with their negative charge to cross the membrane into the cell more rapidly than usual, which causes hyperpolarization. It makes sense. Yay. Glycine's not on there. Is another common okay. Is another common inhibitory transmitter. Fascinating. Acetylcholine is its own thing. Also a transmitter at many ionotropic synapses, has mostly excitatory effects, which have been extensively studied. Excitatory, inhibitory. I haven't mentioned to you. Chloride channels, usually triggered, opened when inhibitory. Okay. That's an image. What's down here? No! The image of the G protein. Damn it. 312A. 312A. Where's 312A? It's this one. Oh, that is rude. How is everyone? Good? Good. Oh no, what happened? Okay, skipped frames is fine. My stream breaking apart is not fine. Okay. Dang. Okay, that sucks. It doesn't show me. And it doesn't really show me Okay, acetylcholine receptor, as it might be seen from the synaptic cleft, its outer portion is red, is embedded in the neuron's membrane. Its inner portion, blue, surrounds the sodium channel. When at rest, unstimulated, the inner portion of the receptor coils together tightly enough to block sodium passage. When acetylcholine attaches, the receptor folds outward, widening the sodium channel. It shows the side of the receptor with that acetylcholine. So that's this image, but it's not allowed. Okay. That's the ionotropic, metabotropic effects in second messenger systems. At other synapses, neurotransmitters exert metabotropic effects by initiating a sequence of metabolic reactions. So metabolic, think of it as your metabolism. It's slow. Okay. Sequence, so metabotropic is slow. Reactions that are slower and longer lasting than ionotropic effects. So ionotropic effects are quicker, whether it's triggered by glutamate, GABA glycine. If it's glutamate, then it triggers an action potential. It opens the sodium gated channels. If it's GABA glycine, it inhibits. It triggers an inhibitory response. And I'm not sure what it does. Oh, okay. It opens the chloride gates. Inhibitory opens the chloride gates. It hyperpolarizes the cell, cancels out an action potential. Interesting. So basically it's like, stop the message, stop the action. No. Cool. Okay. Where was I? Metabotropic effects emerge 30 milliseconds or more after the release of the transmitter. So the excitatory happens in less than 1 millisecond. And they last about 20. Okay. I'm gonna turn on the heater because it's getting cold. Okay. And last seconds, minutes, or longer. Wow. Whereas most ionotropic effects depend on either glutamate or GABA, right, inhibitory or excitatory, metabotropic synapses use a large variety of transmitters, so not just those 3. Okay. They use a lot of other stuff. Okay. Do we talk about G proteins anywhere? No. Excuse me. Okay. When the neurotransmitter attaches to a metabotropic receptor, it bends the receptor protein, enabling a portion of it inside the neuron to react with other molecules. Which was 313. Which is this. 313. So non-stimulated metabotropic receptor, membrane, G protein. Transmitter molecule attaches to the receptor. The receptor bends. G protein activates a second messenger, such as cyclic AMP, which alters a metabolic pathway, turns on a gene in the nucleus, or opens or closes an ion channel. Fascinating. Okay. The portion inside the neuron activates a G protein. There's lots of proteins inside, but this one specifically the G protein. one that is coupled to a guanosine triphosphate, a GTP. That's a new term. I know ATP, I don't know what it means. I'm assuming TP is triphosphate. Sweet Jesus. G protein. G protein. Wow. Guanosine. Triphosphate. GTP. Okay. An energy storing molecule. Protein coupled with a GDP. Energy storing. Energy storing. The activated G protein in turn increases the concentration of a second messenger, such as cyclic adenosine monophosphate, cyclic AMP. Oh my god, terms. Oh my god. Cyclic. Wait, what? Okay. Second messenger. Cyclic AMP. Or adenosine. Yes. Adenosine. Adenosine. That's a purine. Oh. Adenosine. Monophosphate. Okay, I was right. Monophosphate. Oh my god. I'm so scared. Okay. Okay. So it triggers a second messenger called the cyclic adenosine monophosphate inside the cell. So you have a triphosphate and a monophosphate that equals 4. Just as the first messenger, the neurotransmitter, carries information to the postsynaptic cell, the second messenger, the cyclic AMP, communicates to areas within the cell. The second messenger, cyclic AMP, may open or close ion channels in the membrane or activate a portion of a chromosome. What the fuck? Note the contrast. So this is why it takes forever. Wait, okay. Let's read that. An ionotropic synapse has effects localized to one point on the membrane. Yes. Whereas metabotropic synapse, by way of its second messenger, influences activity in much or all of the cell over a longer time. Oh. Okay. What is the purpose? A real life clown, that's what I am. Oh, I sent myself money. Okay. Ionotropic, metabotropic, second messenger, quick. Synapses contribute to different aspects of behavior. For vision and hearing, the brain needs rapid, quickly changing information. Ionotropic, okay. The kind that ionotropic synapses bring. Quick, vision, hearing. You know, I was having this conversation with myself. When do I ever turn off my ears? When do I ever turn off my eyes? Like resting my eyes, obviously to sleep. But your ears while you're sleeping, like when do they rest? I don't know. Cause like I constantly have headphones on or like earphones in. Or like just constant noises all the time. Like when do your ears just like turn off? Because even when you're asleep, like they don't turn on. Like, when do they rest? Our poor ears. My God. Okay, ionotropic. In contrast, hunger, thirst, fear, and anger constitute long-term changes in the probabilities of many behaviors. True. Hunger builds up. Hunger takes time to like have. Thirst, same thing, takes time. Fear. Yeah, we don't have like a quick, like when we're afraid, we don't respond quickly. I mean, the fear might be like watching a friggin' movie and you're scared, you just sit there. Anger, same thing. Anger, I feel, tends to build up. I think anger isn't something that happens just like that. Anger is something that has to build up. Have you ever thought about that? Think about that. Oh my God. Okay. Let's have a topic. Synapses are better suited for that kind of function. So I guess ionotropic is more for like the inside stuff. Tabotropic is more like for the external. Oh, things make sense. Oh my God. Okay, I'm gonna keep that to myself. That was like a epiphany moment. Okay, it's already 10. Oh my God. Okay. So I'm gonna stay on for 15 more minutes because that'll give me a full rounded out 3 hours. But I'm making so many connections in my brain right now. I'm excited. I don't want to leave. But I also need to make sure I sleep early tonight so I can wake up early tomorrow. Okay. I'm so happy I'm reading about this. I finally understand G proteins. G proteins are proteins inside the postsynaptic neuron that bind to a guanosine triphosphate, GTP. Okay. And then that triggers a second messenger, a cyclic adenosine monophosphate. Okay. Yes. Okay. Metabotropic synapses also mediate at least some of the input for taste and pain, some, which are slower and more enduring experiences than vision or hearing. Yeah, like when you get a cut on your finger, you feel pain for a while. Or if like you kick yourself in the shin while you're running. Okay. I'm just distracting myself with connections. Research often described the neuropeptides as neuromodulators because they have several properties that set them apart from other transmitters. Endorphin, substance P, neuropeptide Y. Okay. Endorphins. Whereas other neurotransmitters are released mainly at the axon terminal. Monoamines, I'm assuming? I'm so glad I wrote these because I could just point at you and remember. Visual learning, y'all. Visual learning. Wait. Mainly at axon terminals. Neuropeptides are released by cell bodies, dendrites, and sides of the axons. That makes sense, okay. Whereas a single action potential can release other neurotransmitters, neuropeptides usually require repeated stimulation. Endorphin, substance P, neuropeptide Y. Why? Okay. However, after a few dendrites release a neuropeptide, the release chemical stimulates other dendrites to release the same neuropeptide even without depolarization. Okay, that's just fun information. That is, neurons containing neuropeptides do not release them often, but when they do, they release substantial amounts. Okay. Furthermore, unlike other transmitters, Neuropeptides are not released immediately adjacent to their receptors. They diffuse widely, affecting many neurons in the region of the brain. Endorphins! Okay. Via metabotropic receptors. We shall encounter neuropeptides in several later chapters, especially in the module on hunger. Okay. Did I learn anything about Psyc initiation zone? No. But I learned a lot of other fun stuff. THC. Acetylcholinesterase. Acetylcholine. I think these are all the stuff that I'm supposed to be learning over here. Oh, okay. Wait, how much time do I have left? I still have quite a bit of time. That is 13 minutes? Yes, okay. As you can tell, I talk to myself a lot. It's not weird, okay? It helps you study better. You're weird for not talking to yourself. Oh my god, I'm talking to myself. Okay, I was confusing them. Okay, so let's go back here. Okay, synaptic integration, the importance of synaptic location. Each synapse generates an EPSP of the same amplitude. Synapse, okay, which yields the largest EPSP measured at the Psyc initiation zone. Recall this is the part of the neuron where it is easiest to trigger an action potential. Thus, this is the point at which the output of the cell is controlled. The textbook doesn't talk about this at all. Alright, so Psyc initiation zone. In general, activity at many synapses, it is required to generate a spike at the Psyc. Inhibition will decrease the probability of spiking or reduce the frequency of an ongoing spike train. Let's read the next slide, see if that helps us. Let's say this is one of the motor neurons that mediates the muscle contractions that cause the palatal withdrawal. When it fires action potentials, the muscle contracts. Excitatory, probably ionotropic, maybe metabotropic. It receives 3 synapses that carry tactile information from different closely spaced locations on the paw. Okay. Each synapse results in a small subthreshold excitatory response when activated by touch. If all 3 synapses are active simultaneously, their EPSP will summate at the SIZ and trigger action potentials spatial. If one synapse is activated by several stimuli that are closely spaced in time, its EPSP will staircase and eventually see threshold temporal summation. So this is all just related to temporal and spatial summation. Okay. I've been talking so much. We're gonna move on to spatial and temporal. Yes, sneeze again. 2. Spatial temporal summation. to SRT. Toggle. Okay, my question is, how do EPSPs, how are EPSPs? Ended at 3. Why am I reading what is it? Honestly, I don't know why the top of my head is hurting so much. Like, this is very comfortable. It's required to generate. Myself that I would stop streaming once I hit 3 hours, and I'm 2 minutes out after. Okay, well, I actually got really absorbed in what I was studying, and honestly, I was thinking of just letting the stream continue, but no, I should cancel the stream or like end the stream. Sorry, because it is late. What's it called? I'm having a brain fart. Okay. Thank you so much for being on the stream with me. Thank you so much for studying with me. If I was entertaining at all, yay. Um, I don't know if I will have time to stream tomorrow or Friday. No, I'll probably have time to stream Friday, but my exam is Friday. So wish me luck if you guys have exams this week. Good luck to you as well, and happy studying. It's nice to be productive. Actually, streaming kind of helps me be productive, and if you're also studying neuro brain stuff, good luck to you. And message me, comment, like, subscribe, follow, everything. And hopefully you can catch me on my next stream. All right, bye.